Yeah, this style is...
Good afternoon, everyone. Welcome to our colloquium this afternoon. It's a pleasure and distinct privilege to introduce our colloquium speaker, Professor Aydogan Ozcan. He is a Chancellor's Professor at UCLA in Department of Electrical Engineering, and he's also a HHMI professor at the Howard Hughes Medical Institute. He's also co-leading the Bio and Nanophotonics lab at UCLA, as well as he's a associate director for California Nanosystem Institute at UCLA. He has a long list of accomplishments and distinctions, so I'm just going to summarize that in the interest of time. He holds 31 issued patents, and interestingly, all of them are licensed, which is quite an accomplishment, with 20 pending patents in the areas of nanoscopy, wide field imaging, lensless imaging, nonlinear optics, fiber optics, and optical coherence tomography. He has over 400 peer-reviewed research articles in major scientific journals like Nature and Science, and he has received several awards, including the Presidential Early Career Award for Scientists and Engineers, among numerous other awards. He is also a fellow of the Optical Society of America, NSPIE, and with that kind of summarized introduction, I would invite Professor Aydogan to come talk about democratization of next-generation imaging and diagnostic measurement tools through computational photonics.
Thank you. Thanks for this kind and generous introduction. It's a great pleasure to be here. Thanks for having me. So it's my first time to this beautiful building and a very impressive set of research, of course, but also a very interesting building in terms of its architecture. So I couldn't keep but taking pictures and sharing them in social media as I was entering and walking through the building. Very nice. So in this presentation, I'll share with you some of our work on the use of computation for creating new kinds of microscopes, new kinds of sensors, diagnostic tools. And the theme, the focus area for our work is mobile health, is telemedicine applications, and also global health, trying to bring these kinds of instruments, devices, for resource-poor settings. And in my title, there is an emphasis on democratization because I believe some of these platforms could serve as core pillars for designing next-generation imaging and sensing tools. And that's where I think it's beyond just global health. It also has some implications for higher education, for training, and for conducting certain experiments that would normally be requiring large infrastructure to be done in essentially even the third world. That's essentially a theme. Before I start talking about how the different components that I've briefly described come together, I'd like to start talking about mobile phones as an enabler for some of these emerging techniques, emerging ideas. And I want to start by showing you a comparison of the Moore's law against the megapixel count of our mobile phones and how it's been increasing over the last 10 to 20 years. What you see here is that every 2 years, the megapixel count of our mobile phones, especially CMOS imagers, has been doubling, and it's now reached to more than 40 megapixels. This enables us, through the chips, through the CMOS imagers installed at the back of our mobile phones, to conduct advanced imaging. Cost-effective, field-portable, and very competitive in terms of what kinds of space bandwidth products you can get. And that's quite unique, and it wasn't expected, but essentially it's a remarkable advancement that is helping, in general, computational imaging techniques. In addition to this, mobile phones today are also supercomputers in the way that they're positioned with GPUs and their connectivity. They can actually not only acquire information at the micro and even nanoscale, but also crunch these numbers and create essentially a sensory output thanks to the computational power of these smartphones, especially as teenagers are demanding more and more GPUs for their mobile gaming platforms, this is a trend that I believe is going to help computational imaging tasks to be implemented through mobile phones. All of this advancement, is because of the volume. It's essentially economies of scale helping us. We have about 7 billion, more than that actually today, cell phone subscribers, and this is the driver for these advances that we can take for granted almost. And as you can see, mobile phones are now everywhere, and you can rely on it as a platform to bring some of these new emerging ideas to be implemented almost anywhere that there's a human being. So this is a theme in our lab. We try to tap into this potential to create new kinds of instruments, new kinds of biomedical platforms for conducting measurements relevant to mobile health, relevant to telemedicine, and relevant to global health applications. And on this slide, what you see are some of these interfaces that are 3D printed to convert mobile phones or mobile phone components into advanced microscopes or sensors. So it's now routine to convert the mobile phone into a high-end microscope, bright field or fluorescence. These are some of the early generation devices. I'm not sure if we can call some of these phones as smart anymore, but still, with the recent generation of mobile phones, you can actually push this technology to the level where you can see individual viruses or individual DNA, single DNA molecule that is labeled with a fluorescent tag. So it's come a long way thanks to a lot of these developments coming into play. In addition to mobile phones, you can also look at individual components, especially the CMOS chips borrowed from mobile phones to create standalone designs. So these are microscopes that are based on CMOS imaging technology, 5, 10, 20 megapixel CMOS imagers, but they're working in a non-traditional imaging mode, and they can essentially generate large space bandwidth product images. For example, this microscope that you see here, without the use of any lenses, it can look at about 1 billion useful pixels across a very large field of view, roughly half a centimeter by half a centimeter in area. I'll explain the underlying technology behind this, but just to highlight upfront, it's not just a cell phone. Sometimes it's the CMOS imager that is borrowed from a mobile phone. In addition to microscopic imaging in fluorescence or in bright field mode, you can also convert the mobile phone into a sensor. In this case, you're looking at an imaging flow cytometer, which flushes through a disposable microfluidic device, some cell lines, so let's say in this case blood, and then can look at a movie and process it for looking at counts of cells per unit volume. In this case, you're looking at an Android phone, which looks at a fluorescent signal from urine samples to quantify albumin, which would be important for especially chronic kidney patients with about a few parts per million level of sensitivity. And this next slide, I'll show you some other examples, some other maybe a little bit more recent examples, using fluorescence and bright field microscopy through attachments to a mobile phone camera. You can actually also write an application that will capture and process these images to count red blood cells and white blood cells after some tagging with about the same level of accuracy that you would expect, maybe 5 to 6% compared to an FDA-approved hematology analyzer for looking at cell count. Some other examples are shown here. You can create all kinds of surface chemistry-enabled sensing platforms that the readout and quantification can be done through a fluorescence interface that is read by a mobile phone. It's an earlier generation. Later on, toward the end of my presentation, if I have time, I'll show you some other more recent work for looking at bacteria and waterborne parasites, which also merge machine learning. It's not just imaging, but also some processing done at the back end to give you specificity due to, for example, shape of the parasite that you're looking at. In this example that I'd like to briefly mention, this is actually a nano-particle-based assay, which is looking at, in this specific example, mercury 2 ions, mercury contamination in drinking water, and based on clustering of nanoparticles and the color change that is quantified using an Android application, you can detect the presence of mercury with about 3 to 4 parts per billion, nanograms per mil level of sensitivity. So these are some examples where mobile phones or mobile phone parts have been instrumental for pushing the limits of some of these measurement tools to field portable designs integrated with mobile phones. Recently, there are also other types of devices that everybody's also very excited about, including variable computers like smartwatches and, for example, a variable glass, as shown here with Google Glass. We've also been working on converting those kinds of interfaces to measurement technologies. In this case, you're looking at a lateral flow diagnostic test reader, which captures images in a voice-activated hands-free interface, and everything is done in terms of computation and understanding the quantification of the test strip, color change in response to an analyte is done off the server after some machine learning. These are some examples where maybe the camera interface is not as good as a cell phone. Furthermore, the ambient light conditions would put you in a certain sensitivity limit compared to a 3D printed enclosure. However, there are certain charming user interface advantages of the glass platform compared to a cell phone, and especially voice activation and lens-free operation is unique. And recently, we've converted that into actually a chlorophyll meter in a non-destructive way through transmission of the light through the leaf. You can actually do a similar colorimetric analysis after some calibration to infer in a quantified manner the content of chlorophyll, nicely matching to a handheld or other types of chlorophyll meters available, of course, much more mobile. These give you examples where the designs are significantly more mobile and compact compared to traditional measurement devices, and the advantages coming from consumer devices and the infrastructure around the consumer device itself helps us tremendously to implement these projects. But it's not just about miniaturization. It's not just about cost effectiveness and mobility of these measurement devices. It's also about connectivity. These are connected measurements, and I think that connectivity is also going to be quite important for understanding small and big data patterns by looking at essentially spatio-temporal distributions of these measurements, which is, I think, quite exciting for epidemiology. Imagine being able to ask to an interface a question like, how does a certain disease, let's say HIV, spread over the last 6 months in your neighborhood compared to another neighborhood in, let's say, Los Angeles? These are questions that can only be answered if you have ubiquitous measurement devices that are sensitive and connected to each other. This angle is also quite unique. And I'll emphasize this throughout my presentation with different examples. Now I'd like to come back to talk about microscopy, starting the first example that I gave at the very beginning of my talk, saying that the mobile phone itself, together with the CMOS imager and how it's been improving its space-bounded product, you can actually look at a new microscopy platform integrated with the mobile phone interface. So this is quite important because microscopy is a tool that is almost universal and finds many applications in different fields. In this particular implementation, there are some differences than traditional light microscopes. For example, this interface over here or over the corner, or these standalone devices, they do not contain lenses or a projection, image projection system in the way that we understand point-to-point. They're based on diffraction. They're based on lens-free on-chip imaging. Simply put, they essentially detect shadows of the specimen and reconstruct images in transmission mode from these shadows. As a result, they have certain advantages, also some limitations because of the nature of the design. But the key signature of these kinds of microscopes is the throughput. They will be able to look at very large volumes, very large areas, and very large depths of field. And this will be especially important for looking at rare events. And I'll give you examples of that as a function of space and time. And obviously, these are tolerant to misalignment because of their simple design, which is quite important for the applications that we're seeking for, especially in resource-poor settings. I want to emphasize this much, but the same framework is not only for mobile phone-based, CMOS-based imaging system design. It's also quite competitive if you combine them with wide format CCDs to look at extremely large areas on the order of 10, 20 centimeters square, which is quite large, maybe 3 orders of magnitude, compared to traditional lens-based microscope design. This is going to come somewhere in the middle to just give you a comparison, because whatever I'm going to show you in the next few slides as a platform is independent of the sensor type. I can select any different sensor type depending on my resolution and field of view need. As I've said, these kinds of microscopes are based on imaging of shadows. In a sense, if you have a CMOS imager or a CCD sensor array, you're looking at a certain active area dictated by the number of megapixels that you have and the pixel area. Typically, all of your mobile phones would fall in a range where this silicon chip here would be half a centimeter by half a centimeter. So typically, you're looking at 20 to 30 millimeter square area. And you place the specimen, a sample that can transmit light, very close to it. Typically, H that you see in here is about a few tens of microns to maybe a few millimeters. So it's almost like taking a shadow image of the samples. And if you look at what's happening at the recorded image, you will see these diffraction patterns. These are the shadows, but they are shadows that are quite unique. You can use them as a basis for a cytometer. In this case, you're looking at a whole blood sample. These are red blood cell shadows, and these are white blood cell shadows. So in a simple mode, you can actually forget about the image and look at the diffraction pattern to recognize cells based on a library and do cytometric analysis without any, essentially, image reconstruction. But it's not just a cytometer, but also it's an imaging cytometer in the sense that you can actually reconstruct these images, because these are holograms that are formed by diffraction of light or interference of the scattered light from an object with a background. So essentially, you can take fields of view like this, where there's a crowded region here. Many cells start to talk to each other in a shadow domain, but you can start to reconstruct them to reveal or to backpropagate the optical field after some phase recovery, which I'll describe in the next few slides, to go back to the plane of interest. And you can do this across a very large depth of field. You can focus back to a millimeter, half a millimeter, 2 millimeters, depending on your object. Nicely matching to a conventional microscope objective. So this is a holographic lens-free microscope, but its design is unique, and it's different than traditional digital holographic microscopy techniques that were out there for quite a while. In many ways. First of all, this is incoherent or partially coherent light, both temporally and spatially, meaning that our bandwidth here, for temporal coherence reasons, is not as demanding as a traditional coherent holographic microscope. It can be a few nanometers or maybe 10, 15 nanometers, depending on your resolution need. So in that regard, an LED would be a great candidate for illumination. The second important thing is that the spatial coherence here is not as important. We typically work with a gap between an aperture plane and the sample plane that is a few centimeters, dictated by this tube length. That gives us sufficient spatial coherence at the object and detector plane so that actually almost diffraction-limited information in terms of the scattered field can be acquired or sampled at the detector plane. And this diameter here, the aperture, is very large in our case. Typically, we operate with 50 to 100 micrometer apertures. That's why it's very well suited for a low-power LED, simply aligned to a maybe multimode fiber, 50 micron core, 100 micron core, that illuminates a large area almost uniformly, and that's sufficient for us. So in that regard, it's simple, it's on-chip, and the magnification is unit. It's 1x geometry. Sample field of view equals active area of the sample. So like any other holographic inline microscope, you need to, from those diffraction patterns, reconstruct an image. This can be considered as a twin-image illumination problem, as in the classical holography literature, where you can essentially go back and forth between the real image and the virtual image created by the twin-image artifact, because you don't have the phase sensitivity here. You directly record only intensities. As a result of that limitation, you can actually eliminate virtual image, or you need to eliminate virtual image to get to a final real image. Another way of looking at this is actually purely from the perspective of sampling or measuring a field's amplitude that is missing phase. So you can actually purely look at this framework as a phase recovery problem and try to find the phase of the optical field whose amplitude we've measured. And this is a better framework for some of the work that we do, and we'll stick to this one, and I'll show you some examples of how we do this. In the most simplistic way, you can actually go back and forth between the hologram plane and the object plane. By throwing random phases, you will have objects that are reconstructed incorrectly, leaking energy outside of a normal region of interest or an object support. And that energy can be minimized iteratively by correcting your phase at every iteration and enforcing the amplitude measurement and the object support. This is not exactly what we use currently because we do not like to enforce object support in our reconstructions. Later on, I'll show you results where there are other degrees of freedom that we can enforce for this type of an iterative algorithm to converge for any sample of interest where you cannot sometimes put a relevant object support. But this is a good start to understand that you need to do phase recovery, and we always do this iteratively. And typically, these are fast algorithms that rely on FFTs. And GPUs help a lot because different parts of your field of view can be distributed to different cores. Under this simple design and phase retrieval at the backbone, you can create a very compact and simple microscope by a single LED, low power, battery powered. A nice CMOS imager, 510 megapixel, typically having a pixel size of 1 to 2 micron. Place the sample from the side, look at the transmission images. Measure the amplitude, recover the phase quickly, and you can go back to object plane to retrieve amplitude and phase images of the specimen. Even with the simple geometry, you can actually work this platform as a specific cytometer for even objects that are very difficult to differentiate under a light microscope unless it's labeled. You can actually do surface chemistry to capture different subpopulations of cells and start reconstructing them and counting them. In this example, we're focusing on lymphocytes, CD4 positive and CD8 positive lymphocytes, with the hope that for an HIV positive patient, we can monitor the depletion of CD4 lymphocytes with respect to CD8s. And that's where these kinds of devices could also be used for monitoring of the progression of a disease like HIV through cell counts. This is a good example where resolution is secondary because your specificity comes from surface chemistry. In this case, this spot here is specifically functionalized with anti-CD4s, antibodies, and in this case, anti-CD8. And that gives us a nice ratiometric analysis of how well the patient is doing. This was one example in terms of where resolution doesn't matter if you bring specificity and sensitivity, depending on your chip design or fabrication. In this simple form, you're looking at a very lightweight design, about 40 grams in this case. And the resolution here is dictated by the pixelation. In this unit magnification geometry, one major advantage that we have is the field of view. 1x geometry is beautiful because the active area, that half centimeter by half a centimeter active area, is my object field of view. In this case, it's about 24 millimeters square. But effective numerical aperture of these kinds of reconstructions in this simple design is fundamentally limited by sampling, spatial sampling dictated by the pixel size. In this particular example, we're looking at about 0.1, 0.2 numerical aperture, effectively created by about 2 micron pixel size in this native CMOS sensor. This is useful in a sense for mobile microscopy applications, but there are a lot of other applications which demand higher numerical apertures, higher resolution, and without trading off the field of view. So the next step that we've that's one lady, yes. So the next step for us was to push this platform to the next step in terms of improving the resolution without sacrificing its design simplicity, cost effectiveness, mobility, and field of view, meaning 1x geometry, active area. For this, we've borrowed some mathematical frameworks from especially photography literature. And we use pixel super resolution to essentially digitally embed more pixels to our native holograms. A quick disclaimer, super resolution is first used in photography literature before fluorescence microscopy started to use it for beating the diffraction limit of light. So in that regard, this is still diffraction limited imaging in the sense that we're not bringing evanescent wave information here. We're strictly working with traveling spatial frequencies, not evanescent. And the resolution enhancement here is through eliminating pixelation. To do that, we've complicated the design a little bit. Essentially, instead of a single LED, we have some now 20 LEDs. And we turn them on and off one by one. The sample is as before in front of a CMOS. You capture undersampled holograms, one at a time. What is different here is that there's now a time sequence where the light source shifts, as a result of which the undersampling pattern also shifts, because the shadows or holograms start to shift. So the framework then is, given a set of undersampled holograms, how to retrieve the pixel super resolved hologram so that some of these frames that are washed away in details could be reconstructed back. This is the same thing that we do based on an optimization. Basically, we define a cost function and try to come up with a super resolved hologram which effectively has much smaller pixel size compared to the native pixel size of the sensor that is compatible with all these subpixel shifts. One thing that's important here is that you do not need to know the subpixel shifts in advance. It doesn't come from the hardware. It's actually understood from frames by essentially cross-correlation. And that means these kinds of devices do not need to be calibrated, which is very important because calibration in field settings will be very difficult under different temperatures. So it's self-calibrated through reconstruction. At the end of this process, some of these fringes that are normally washed away because of pixelation start to come back. Those rapid oscillations that are retrieved here are corresponding to higher numerical apertures, which led us to reconstruct higher spatial frequencies that are normally washed away. This is a good example, which is coming from a very old sensor, just to show the proof of concept. It's a 5 megapixel imager, which I believe none of your cell phones currently have. A single reconstruction would not yield the resolution that you demand, but with super resolution, with pixel super resolution, you can reconstruct both amplitude and phase of these objects. And you can still put this together in a field portable design. This is about 150 grams, this initial design. Again, this is not a very competitive one because it's using an old sensor. It's a 5 megapixel, 2.2 micron sensor. But it still is pretty nice for looking at, in the phase reconstruction channel, malaria infection. This is a red blood cell, and these are plasmodium falciparum that is stained, that gives a stain to kind of give you contrast in the phase channel. And this is the comparison, which gives you essentially a nice feel of how it compares with a traditional light microscope. If you were to use a state-of-the-art sensor, not that older generation that I've shown you, you can actually create some very competitive images across very large fields of view. This is showing a sensor which has about 20 millimeter square active area, 4 by 5 millimeter in width. And it has a pixel size of about 1.1 micron. Through pixel super resolution, you can actually reduce the effective pixel size to about 100 nanometers and increase the space bandwidth product from about 10 megapixel native sensor to about 1 billion pixel. And resolve half-width lines of about 225 nanometer, which is using green wavelength. So about 0.9 to 1 numerical aperture can be achieved with these kinds of platforms. If you want to push the resolution further, you can also have some other tricks. One trick that we've utilized is to play with the illumination angle. Vertical illumination gives you a certain band that is diffracting. As I shift the illumination angle, some of the evanescent waves or some of the higher frequencies that are normally going to be missed away because of the pixel architecture will start to shift to lower frequencies. And vice versa, DC will start to shift to higher frequencies. This angular diversity, combined with pixel super resolution, gives you a frame to about double the numerical aperture of this system, which ultimately is limited by the refractive index of the medium. A lot of times we use some oil between the sample and the sensor to even enhance that. And this kind of a platform gives us stability in phase reconstruction. We have redundancy in the frequency map because different angles give you some overlaps. But if you synthesize all these different angles, you essentially increase the passband of your spatial frequencies from a circle like this in spatial frequencies to a circle like that due to synthetic aperture. And this gives us a numerical aperture that is larger than, in this case, the refractive index of air, which is essentially still limited less than 2 times the refractive index of air in this case, mostly because of signal-to-noise ratio. And this also helps us with phase reconstruction stability for complicated objects like tissue samples. What you're looking at here is a tissue sample, about 20 millimeters square field of view. And you can zoom in to our reconstructions that show you how it compares with a traditional light microscope for all these features. It's quite interesting. In this case, the colorization is coming from individual sequential illuminations at different red, green, and blue wavelengths. Because this is holographic, it's more convenient for us to do colorization after 3 sequential illuminations. In terms of a recent state-of-the-art field portable implementation, this is what you are looking at. It's essentially a pixel super resolution microscope that has some LEDs here. It has also some color so that we can do colorization digitally. One additional degree of freedom here that I haven't mentioned so far is modulation of the gap between the sample and sensor. That modulation, just like the super resolution aperture modulation, doesn't have to be repeatable. And we do not need to know that because we can actually do autofocusing to understand the delta Z. But the advantage of Z-scan is, without any assumptions, you can have redundant information at different heights from the sensor surface so that I can take, for example, M measurements, typically M is 3 to 5, and throw a random phase to any plane and create a complex field that is very naive at first. But as you propagate and go back and forth among different planes, you refine because you know the measured amplitude and you retain the phase. It's always about retaining the phase and feeding a measurement. This is very robust. And if you combine this with corrections of plane angular mismatch between different planes, it even becomes more aggressive in convergence to essentially the correct solution, even using a simple field portable design. And you can take holograms like this and start to essentially go to deeply subcellular resolution through phase recovery. This is a good example where you're also looking at a breast cancer tissue. Typical field of view of a 20x objective is shown here. Maybe a 40x, a higher resolution is shown here. And these are the zoomed-in images showing these subcellular features. To understand how well we preserve details, we've done a blind study with a board-certified pathologist and gave her some of our images and some of the traditional lens-based microscope images to see how the diagnostic value for this case of breast cancer diagnosis is preserved. And we've got about 99% match to a gold standard microscope comparison, validating that you retain the pathological information and spatial frequencies related to that. This is another example. It's a Papa Nicolau smear, pap smear. It's used for early screening of cervical cancer. Advantage is the same, a field portable platform for looking at large fields of view and preserving also the details. And one important advantage is you can actually take these images and send them to a remote pathologist that may be a few hundred kilometers away. And you can also do depth focusing digitally after exposure, which gives the pathologist an advantage to do out-of-focus examination, because a lot of times these kinds of samples have depth to it. So some features are out of focus because of cells wrapping onto each other. So this depth focusing is also quite exciting. So far, I've been focusing on CMOS imagers, all of which are being used in our mobile phone cameras. And this is the signature over here, a scale bar of 1 millimeter and half a centimeter by half a centimeter, typical field of view. That is the signature of a CMOS of a mobile phone. You can also do CCDs, and then the scale bar becomes a centimeter, and that's where the advantage of a CCD is. You can look at a 10, 20 centimeters square in a snapshot. Of course, that comes with a resolution loss because mostly, generally speaking, CCDs have larger pixel size. So super resolution helps, but still cannot give you deeply sub-micron resolution, as in the case of CMOS technology. But still, from very pixelated images, you can go to sub-pixel holograms, reconstruct. These are sub-micron tails of human sperm that can be reconstructed. In this case, the resolution is about 2 micron, which gives you about 1.5 gigapixels. One important distinction that I'd like to make at this point is this is a platform that is independent of the sensor chip. Depending on your resolution and field of view, you can select different amounts of megapixels, different pixel size. And more importantly, in classical microscopy, depending on your objective lens, there's always a relationship, a trade-off between resolution and field of view. They're tied to each other. You have to sacrifice one to get the other improved. This doesn't apply here because field of view and resolution are independent in this kind of a unit magnification on chip geometry. If you give me more megapixels and smaller pixel size, I can improve both pixels, both resolution and field of view at the same time. It's quite important, and that's the reason why I've shown or started with that Moore's curve, because it's very important for us. That drive is helping us improve our technology without doing much. We're riding on that Moore's curve. So far, I've been talking mostly about pixelation, how to improve the resolution of the system to the diffraction limit, while retaining the advantages of the platform. The other important feature of an imaging system like a microscope is its sensitivity. You can claim a resolution that has a very large NA, but your detection noise will determine what kinds of small features you can ultimately detect. So resolution is an inherent function of signal-to-noise ratio in any imaging system. This is true for this kind of a modality too. To see smaller things with this kind of a geometry is even more challenging because it's trying to see the moon in a sunny day. Your detector is overwhelmed by the background light. To solve this kind of a problem, we borrowed tricks from self-assembly, and we created self-assembled nano-lenses around particles, nano-particles, to essentially increase their signal-to-noise ratio. This is the work that one of your new professors has pioneered, and this is actually the work that, as a postdoc, he did before coming here to push the limits of this system to the level where we can see sub-100, sub-50 nanometer particles. This is actually a field-portable design that Dr. Ewan McLeod, who is right in the middle, has created. It's actually a platform which, over the same field of view, can look at about 40 nanometer, as small as 40 nanometer particles, as large as a few hundred microns, a huge dynamic range in the particle size, and particle density, which is quite unique. In terms of looking at nanoparticles or microparticles in air, water, this kind of an interface is quite unique because it can handle a large dynamic range in size and density in a field-portable implementation. So I won't go into the details of this platform because I know that some of you are very well familiar with this platform through UN, and if you are interested, I would recommend you to follow up some of those ACS nano papers. And this has a good sizing accuracy, about 10 nanometers across a large dynamic range. So the same platform can be combined with surface plasma and resonance-based sensing devices to convert this into a microarray or SPR-based plasmonic sensor. In the interest of time, I'll skip this work as well, but this is in collaboration with an EPFL group to demonstrate sensing of monolayers of protein binding events through transmission and phase retrieval without giving away the advantages, especially in terms of field of view and throughput. So far, we've been discussing microscopy approaches that are based on computational imaging on a chip. for mostly static objects. I didn't discuss time in my current so far presentation. And we were looking at static objects that potentially will be tossed away or stored for further diagnostic analysis, like any other system. But on-chip microscopic imaging with these kinds of partially coherent fields is also quite unique for tracking micromotion in extreme throughputs. I'll give you examples of that and the results that this kind of platform enabled. To track a micro swimmer in a large volume, you place it above the sensor with a few hundred microns, and imagine that there's a tiny pool that has a base area of, let's say, 20 millimeters square and a height of about 0.5 millimeter. There you go. You have about 10 microliters of volume there, which is giant. And these things are swimming freely, in this case, human sperms, enjoying this giant pool for their own swimming. Our goal is to track their motility with sub-micron 3D localization accuracy per sperm head, but not just a few of these sperms, thousands of them in this entire volume. A traditional light microscope has a mismatch in terms of observation volume. And a microswimmer or a sperm would quickly pass by the focus point of an objective lens. And this is where this kind of a platform is quite unique. What you notice here is that this is not the same design as before. It's slightly different in two ways. There are two illumination sources that are simultaneously on at two different angles and two different colors. This is by design to differentiate vertical projection of the hologram from an oblique projection, so that we can pinpoint not only XY, but the Z motion. As you play with the height of a sperm, the separation between the red shadow and the blue shadow will be modulated. And that modulation correlates with delta Z. This is a very nice design to separate also projections that are simultaneously acquired, because blue and red are far apart from each other. So only at the correct angle and wavelength combination you can have contrast. And I emphasize that this is simultaneous acquisition, which means they eat away their contrast. Blue hologram eats away red contrast, and vice versa. By having the correct reconstruction parameters, you can see the volume in two perspectives and pinpoint to XYZ position with sub-micron total localization accuracy, with the same throughput across the 10 microliters. This enabled us to look at extremely interesting patterns because of our throughput. This is a human sperm that is in vitro conducting a helix. It's about a second in motion. It does about 10 rotations per second, very tight rotations, 2-3 microns in diameter. This happens only 4 to 5% of the time. And as you can see, it's quickly moving out of a certain depth plane. That's the reason why this has never been observed, this kind of a motion, using a traditional light microscope, because of the focusing problem as well as the low throughput. This is another one. It's a hyperactivated human sperm. It has more energy. It's slightly irregular, but still a nice, beautiful helix. This platform can look at not just a few, but 1,000 or 2,000 sperms in parallel and record, at the end of the day, holograms that you can process to create trajectories, thousands of them. And you're looking at a crowded scene like this, where in this case there are about 1,500 of them, tracked across 11 seconds. And you can generate many of these across a few hours of an experiment. And we've done this with many sperms. There are 24,000 dots here, each corresponding to an individual trajectory of model sperms. Out of these model sperms, only 4 to 5% are helical. And what's quite interesting is that out of these 1,000 helical sperms, over 24,000 model sperms, 90% of them are right-handed. So they prefer a right-handed helix over a left-handed one. And that's an interesting observation that essentially is only enabled by the throughput of this platform. It takes 96 out of 24,000 to get to this kind of a result. We do not know the reason for this. We have speculations about how this is enabled, influenced by the dislocation of the nuclear material, nucleus of the sperm, how it's off-axis from the rugby ball type of sperm head. And as there is momentum coming from the flagella, essentially that off-axis geometry could give you, together with the surface morphology of the sperm, could give you essentially differences in rotation preferences. And since then, we've been playing with this platform with different things. We've got some very interesting results unpublished, which can even take this to the next milestone. But over the last year or two, we've looked into other species beyond sperm, human sperm. In this case, looking at horse sperm, we were able to discover some new patterns. These are ribbons, chiral ribbons, we call them. So you have a sinusoidal pattern, zigzag pattern on a plane. Very simple modulation. But that plane itself is twisting in the form of a ribbon or a twisted ribbon or a helical ribbon. This is actually a horse sperm that we're recording over 4 seconds. And it's connected to this beautiful helix, as you can see here. It's soon going to rotate and give you the perspective of the sheet and how it's essentially conducting this sinusoidal pattern. Obviously, this is a quite regular pattern that can be mathematically formalized with a beautiful function. But at the same time, it's the first time that these kinds of regular patterns have been observed in a microswimmer system. It's another one. It's also the same horse species. And this is a twisted ribbon, which I'll fast forward, giving you also the different perspectives. This one is also an interesting function. It's a minimal surface. It exhibits zero net curvature. It's helicoid. And interesting thing, minimal surface functions came across in another place when we were doing nano-lenses to see individual nanoparticles. I haven't described this, but essentially catenoids form another group of minimal surface functions which also exhibit zero curvature. And I think a catenoid was the second minimal surface that's proven beyond a plane, simple plane. These are interesting things that are enabled by the throughput, computational power, and also the accuracy of these kinds of high throughput imaging systems. So I want to come back to this this slide where I started. A lot of what I've shown is really utilizing this kind of a growth curve to improve the performance of these platforms. And we've been really enjoying, from 2005, 2006, the next generation of sensors that are embedded in our mobile phones to push the boundaries of this platform. And in that regard, I'm really grateful for consumers replacing their phones every few months. That's essentially great for what we do. I want to share with you a few examples in the other domain, fluorescence microscopy and sensing domain. Bright field is great, but it has its own limitations, especially when it comes to specificity. Fluorescence microscopy and sensor designs utilize beautiful chemistry at the backbone to bring your imaging system the comfort of the specificity that you bring with that surface functionalization or surface chemistry or bioconjugation. And that's where I think fluorescent designs that are also utilizing this kind of a mobile interface would be quite important. In this design, you're looking at a dark field fluorescence microscope that can see individual viruses with a very simple design which utilizes the camera phone with its embedded lens. And then we have another lens which is flipped to cancel out some of the spherical aberrations. And then it gives you a low magnification but a very high signal-to-noise ratio dark field microscope, to the level where you can actually see these are nanoparticles that are being imaged compared to SEM, and these are actually single viruses that are labeled through their glycoproteins. These are the envelope proteins that are being labeled here. These are human cytomegaloviruses that are individually imaged by a mobile phone, and this is a comparison image captured by a confocal scanning microscope. And you can utilize these kinds of platforms with the correct surface chemistry for viral load measurements, for looking at essentially densities of certain specimen in your sample. Recently, we pushed the same platform with some server backhand, some application that communicates with a server to see individual DNA molecules. Actually, this is an SEM image of stretched DNA molecules, and this is a cell phone image showing the same field of view. Resolution is not high, but the contrast is good enough to see individual DNA molecules, which, it's about 2.2 nanometers, the width of the DNA. And every base pair is about getting 5 to 7 fluorophores in that compact space. And the sample prep is pretty simple here. It's actually based on surface friction. They're not oriented well, but who cares, because it's an imaging-based system. These images are sent to a server, which, based on the point spread function of the system, essentially is trying to measure the length of the DNA. We care about the length of the DNA because we want to bring gene-specific imaging to this platform. Our length sizing accuracy is about a kilobase pair. 1 kilobase pair for 10 kilobase pair and longer DNA segments, we can actually measure the sizes. So it will be useful for actually bringing in gene-specific measurements. And some applications that we're currently pursuing is actually to understand drug resistance by labeling certain genes and then trying to understand the copy number variations. Let's say you have malaria from your blood. We can understand if the species that infect you has a certain gene more than replicated in the backbone, more than, let's say, 3, 4, which would mean that we need to change the drug to another drugs for handling the drug resistance issues. So this combination with machine learning and server backend processing can be applied to many different things. This is actually a Giardia detector that's working based on fluorescence microscopy. It's the same idea. Basically, you have a fluorescence microscope there, but you also build a cassette that screens large volumes. What you see in this cassette is a microporous membrane. It has about 5 micron pores, which you can pour in 10, 20 milliliters of water. It will capture a certain size, in this case, Giardia cysts. These are cysts that infect a lot of the US cases for waterborne related diseases are due to Giardia and cryptocysts. And in this domain, this is one way to capture across a large volume and be able to recognize them through their unique shape. This is actually a microscope image, cell phone microscope image. If you zoom in, you would be seeing these rugby ball type of Giardia cysts that are sent to a server for quantification, and the results come back. This has achieved about a cyst per mil limit of detection, which is pretty good for something like this quick. We're now pushing the sensitivity further for this platform. And finally, I'd like to mention that you can bring some other interesting tools for spatially multiplex sensing. What you're seeing here is a microplate reader. Microplate is essentially a plate. It can come in different formats. What you see here is a 96-well plate microplate. Essentially, it's a standard format for a lot of the clinical tests, a lot of the ELISA tests that for the samples that you give in a clinical lab. They're using this kind of platform for looking at color changes or for looking at fluorescence to quantify the biomarker concentration or give you a diagnostic decision. Of course, as you can see here, the size is very large. It's a few centimeters by a few centimeters. Typically, clinical instruments use a scanner and have a scanning optical system to look at these individual wells one by one. We've created a system that is based on fiber optics and using the cell phone as an app to acquire these images. The idea here is that by using each well assigned to a multimode fiber, you can look at the transmission intensity, and the fiber bundle here reduces so that a simple optical interface can image the entire 96 well plate. And then we do machine learning on this and understand the concentration of each well corresponding to each fiber optic cable. This one is done off the server. So the main function of the device, the handheld device, is to capture transmission images of this fiber optic table, which again corresponds to these wells. And then we get back the clinical values through the same interface. We've tested this with actually several FDA-approved tests with more than 1,000, in fact 1,100 patient data at UCLA Clinical Microbiology Lab, and we've achieved very good results. With machine learning, we were about 99% accuracy compared to an FDA-approved test reader for measles, mumps, HSV humans, simplex virus 1 and 2. for these 4 tests, which is great for especially immunization campaigns. Imagine you want to screen a village to understand how well they're immunized. Right there, do immunization if needed. This kind of a platform would be unique because now you can actually have hundreds of people within a few cassettes be screened, and the results will come within just a few minutes per cassette. So it's essentially server transmission, and then under 30 seconds the results come back processed. So how much time do I have? 30 minutes. Okay, cool. So before I conclude, I'd like to briefly mention and talk about another aspect that is quite intriguing, and that is the mismatch between high throughput sensing and imaging technologies and diagnosticians or experts who are supposed to look at those images and label them. So this big data is interesting. If you can generate large quantities of data from everywhere in the form of, let's say, gigapixel images, who's going to look at this data and who's going to label them? That's the bottleneck that we're facing. That is the bottleneck between human expertise and the digital microimaging technologies that are increasing their space bandwidth products rapidly. Unfortunately, the humans don't. And that's a major problem. That's a mismatch. We will be buried under data, under images, lots of images. And that's, I think, not a good idea. So to point to this problem and potentially offer a solution, we've created a platform for crowdsourcing of image labeling and diagnosis through images. We created games that essentially took microscopic images of red blood cells taken with different types of microscopes. And the goal is to look at these cells. Each one of these individual rectangular boxes here is actually a red blood cell microscopic image. And just like a diagnostician, your task here is to say which ones you believe are infected. Even if you're not an expert, you can help us with this kind of an interface. This was a unique interface. You can still play it, actually. If you go to biogames.ee.ucla.edu, you can play this game. It's available online. We had different stages of this thing. There's something smart here, which is every screen that you have, we have images that we're trying to understand the correct label. But there are also images that we know the labels for, which means every time you're clicking on a single page, we also evaluate your diagnostic accuracy. We assign you false positive and true positive rates based on the known image that we hide within a screen. And there is a nice mathematical interface that converges from your clicks to the correct diagnosis. Initially, we launched this platform more than 3 years ago now. And now it's played by more than 80 countries, thousands of users generating lots of cell diagnoses. And every one of those is actually a user that has been helping us diagnose malaria. The idea here is we treat this as a noisy telecommunication network. So every gamer is a repeater in the network, and it's broadcasting 0 and 1. 0 is let's say it's a negative cell, and 1 is an infected cell. There's noise in the system which we characterize through their prior probabilities by the hidden images, and there's a decoder that essentially takes maybe 5-10 gamers together and tries to come up with the correct diagnosis for every cell, every image that we have unknown waiting to be diagnosed. This platform got very strong in terms of what it can do. With 20 undergraduates, about 20 undergraduates from School of Engineering, no training on malaria whatsoever, we were able to collectively create a diagnostic accuracy at the single cell level that is about 2% of the accuracy of an expert diagnostician. Actually, about 1.5% of an expert diagnostician who went through the same image set. One of my colleagues from UCLA Clinical Microbiology. And this gets very powerful because it's using the wisdom of the crowd. This is quite interesting because you can also apply this to a crowd of experts. You can, for example, create a system where you're not playing with undergraduates anymore. You're playing with medical experts, professionals, but you do not know their training. And you can actually create a system that beats the best of the group, which makes efficient diagnosis. Later on, after the undergraduate test phase, we took this platform to experts, to professors who make a living out of malaria diagnosis. One thing that we have observed is that experts disagree with essentially their colleagues. There's a lot of disagreement on what they see, positive or negative. And they also are not self-consistent. We've given the same expert the same image 3 times by rotating it. After a few hundred other images in between, they were not self-consistent always in their own decisions. So this gave us a nice statistical framework to create actually a global library of cell images that are very well characterized by the consensus of the experts. And actually, our goal was two phases, 2 different directions. One, to digitally create diagnosticians by combining a group that is not competitive individually, but when they get together, they become super pathologists digitally. And the second, to create training tools for evaluation of diagnostician quality. This phase is now recently implemented. Now you can actually go to biogames.ee.ucla. We're not trying to learn anything from the crowd anymore. After millions of diagnoses, we know very well what every single image in our library is in terms of being positive, being negative, being questionable. Questionable means that image in itself cannot be used for diagnosis. It's something that's also very important. So based on these 3 labels, we're now giving a framework for training and for education of diagnosticians for the specific task of malaria diagnosis by essentially giving them quizzes that are each composed of 500 cells. And at the end, we give them a score. And we also teach them their mistakes in the form of false negatives and false positives. One thing before I conclude, the top diagnostician here, there's also a leaderboard. This is an old leaderboard, but the top diagnostician is actually a 12-year-old Korean girl from a middle school in Korea. We've been tracking, under IRB approval, we've been tracking the performance of middle school kids from Korea. If you ask why Korea, I'll answer it in the Q&A. From Korea, we've been tracking the progress of learning of middle school kids in large numbers. And we've seen that some of the kids, in a matter of 2 weeks, from a negative score, lots of penalties, they go to a very high score. A high score that is better than my co-investigator in a grant who is running our clinical microbiology lab. I forced him to play this game once in front of me, and he got about 60, 65. And these high school kids, middle school kids are about 70 to 80 now in their scoring. It gives you a great example of how some of this learning could be quite interesting and exciting to observe. By the way, some of the middle school kids never learn. They stagnate, and so it's what is the difference between them? How does it correlate with GPA? It's something that we're now trying to understand. So to conclude, I've described our work on the use of computation to create new kinds of imaging and sensing tools, with an emphasis on applications in mobile health, telemedicine, and global health, where the idea is to create competitive designs for these devices that would match the performances of traditional instrumentation that we have in advanced laboratories, so that some of these tests, measurements, can be done in extremely resource-poor settings. That's where telemedicine, that's where mobile health and global health come together. Democratization of devices and instrumentation and measurement tools come together because of the fact that some of these tools, beyond biomedical applications, could be very well integrated with curricula, higher education, even high school, to bring some of these measurements to the hands of essentially undergraduates or high school kids. It would be wonderful to see an undergraduate student or maybe a high school, middle school kid early on see an individual DNA that is stretched, which I haven't seen until this research, till this age. With this, I'd like to conclude and thank you very much for your invitation and your patience.
Thanks for that great talk. It was really exciting to see some